Hello and welcome to my lab experiment video. This will be about the rate of reaction in a catechol oxidase reaction at different temperatures, created by myself, Tyler Karate. So we'll just be going over the outline quick. So we're going to have an introduction, hypothesis, experimental design, results, conclusions, and references. So, first, a quick introduction to the oxidase reaction. When exposed to oxygen, catechol oxidase will oxidize the catechol enzyme and produce melanin, which causes browning in fruits and vegetables. So if you look at the picture on the bottom right there, it'll show you a potato that has not been browned yet, and a potato where the catechol oxidase already oxidized the enzyme and causing it to brown. This happens in all fruits and vegetables because the catechol oxidase is only found in plant cells. So what this enzyme does is it will form a catalyst that will help aid the reaction, and it can cause it to be quicker. For this experiment in particular, there will be 4 different temperature values: 5 degrees Celsius, 25 degrees Celsius, 75 degrees Celsius, and 100 degrees Celsius. The goal of this experiment is to determine which of the optimal temperature for this catechol oxidase reaction in the potato. It's to determine what is the optimal temperature out of the 4 temperatures that are given. So, my hypothesis: given that most reactions, when a temperature is increased, the rate of the reaction will increase. What that means is basically, as the temperature goes up, the rate at which the reaction will occur will also increase. So as the temperature is increased, then the rate of the reaction will increase up until the enzyme starts to denature. When an enzyme gets too hot or too cold, it will start to denature. So I hypothesize that somewhere between 25 degrees Celsius and 75 degrees Celsius is the optimal temperature. If you take a look at the image at the bottom, it shows a normal enzyme and then a denatured enzyme. So what happens when an enzyme denatures is it loses its shape. Enzymes are made to have a certain shape that fits their substrate perfectly because it is unique to that substrate. So if an enzyme denatures, it no longer can fulfill the function it's supposed to. So if it gets too cold, it'll denature, and if it gets too hot, it'll denature. Somewhere in between 25 and 75 is my prediction, or my hypothesis. So the rate of reaction at different temperatures. This is my experimental design. So first off, a graph will have to be made showing how the different temperatures will affect the rate of reaction. The independent variable will be the temperature in degrees Celsius, while the dependent will be the percent of enzyme activity. After the experiment is graphed and the optimal temperature is found, the percent error will be calculated to find the exact optimal temperature of the reaction in potatoes. So I have a little model graph there at the bottom. That is a graph that will show what in the results, what your result graph should look like. How as the temperature increases, the percent of enzyme activity will also increase, and then it gets to the optimal temperature. Once it is at that optimal temperature, it will start to decrease because it will denature and it will no longer fit in its substrate property, and the enzyme activity will go to 0. Like it shows in that graph. I don't know what enzyme this is for. This isn't the catechol oxidase enzyme, but once it hits around 50-ish, it'll start to denature, and once it gets to 60, there's 0 activity. That is what will happen. So, the results. If you take a look at the graph there on the right, this is what the graph should look like given the data values that were given in this experiment. So it shows that the optimal temperature was indeed 25 degrees Celsius. It shows that once it gets to 25, as it starts getting even hotter, it will continue to decrease. apologies, until it gets over to 100, where the percent of enzyme activity then hits 0. So the optimal temperature found is 25 degrees Celsius. The percent error is 28.6 degrees Celsius, because the actual temperature for a catechol oxidase in potato, the optimal temperature is 35 degrees Celsius. So, given that the 4 temperature values, it turns out that 25 degrees Celsius would be the closest to the optimal temperature. Again, it shows that temperature is the independent, and percent of enzyme activity is the dependent variable. So, in the summary here, my conclusion is that my hypothesis that the optimal temperature would be between 25 degrees Celsius and 75 degrees Celsius was correct. In this experiment, we learned how to graph an enzyme rate of reaction graph and how to find the optimal temperature. I feel that my experiment opens up the possibility to have other experiments that look to see if optimal temperature will be different in other types of vegetables and fruits, or if they're all the same. Like, will the optimal temperature of a potato, which is 35 degrees Celsius, be the same as an apple, or as a banana, or as a strawberry? Maybe that these different fruits or vegetables will have different optimal temperatures. The rate of reaction will be different. Something along those lines. But yeah, then we have my references. So thank you for watching. This has been my catechol oxidase reaction lab report by Tyler Karate.